Hello, my name is Elizabeth, and this is Erin, Adriana, and Maya, and our experiment is called Planarian Re-generation in Microgravity. We are all rising ninth graders from Kent Place School in Summit, New Jersey, and our teacher facilitator is our science teacher, Wendy Hall.
After doing extensive research on both microgravity and planarian, the experimental question recreated was, how does a microgravity environment affect the process of cellular regeneration in Dugesia tigrina? Since we wanted to show the planarian's relevance to helping humans, we then created a follow-up question, which was, how this pertained to Homo sapiens in a similar environment.
The first thing this experiment would do is provide proof that regeneration is possible in space. It is important to know that the proper cells will be able to repair an astronaut's cut in microgravity the way it does in gravity. If those skin cells cannot regenerate the skin around it in microgravity, it would be a risk for astronauts to conduct any experiments that could possibly get them injured. Because of the possibility that the skin cells will not regenerate, running this experiment on planaria would provide research about regeneration without risking human health. This experiment could also help scientists infer that regeneration cells need gravity to function. If the regeneration process does not occur, then scientists can infer that cells need gravity to carry out their processes. If they do regenerate, then scientists can infer that gravity has no effect on the regeneration process. Once the scientists narrow down what triggers regeneration, the information gathered could be used on a larger, more complicated cell, such as a human cell, to find similarities and differences. Finally, if there are similarities and differences between the regenerative planaria ones and the human cells, it would further studies of the possibility that humans could harness the power of regeneration in the future, which would be a revolutionary discovery that could help people all around the world.
The planarian might seem ordinary, but its regenerative qualities allow it to do some extraordinary things. One of its most fascinating qualities is its ability to reproduce asexually. Asexual reproduction is the process in which offspring is produced from only one organism and only inherit that one parent's genes, making the new organism genetically identical to the old one. The planarian reproduces asexually through fission, meaning it splits itself in half and then regenerates both missing halves. Not only that, but the planarian can also regenerate itself spontaneously if it becomes injured. This is what made the planarian such a good candidate for our experiment. Through our research, we discovered that the reason the planarian can regenerate itself like this lies in its cells. The planarian is made up of cells that can change into any kind of cell depending on which ones are necessary at a given time. For example, if a planarian has its tail cut off, its cells have the ability to recognize this and then turn into cells that are needed to create a new tail. The planarian cells are very similar to human stem cells. With that being said, humans can also regenerate cells. For example, when we get a minor cut or wound, our bodies have the natural ability to repair themselves. But we have reason to believe that this would not be the case when the cells are exposed to a microgravity environment. We think this because in microgravity, cells degenerate, which is called cellular atrophy. One example of this is astronauts' bones and muscles have been shown to degenerate when exposed to prolonged periods of weightlessness. This happens because the muscles and bones are no longer fighting against the gravitational pull, which causes them to be used less and then degenerate. Since this happens to cells in microgravity, we would like to test how the planarian's ability to reproduce asexually will be affected by a microgravity environment and how that will pertain to humans when placed in a similar environment.
The following is the picture of an FME tube that we would like to send into the microgravity environment. Volume 2 would contain 5 brown planarian that we would be using and 2.8 milliliters of fresh spring water for the planaria to live in. Volume 3 would contain 1 gram of now food liver powder, which would be the planarian's main food source during their time in the ISS. Volume 1 would contain 10% of neutral buffered formalin, which would be used to terminate their planarian on the descent back to Earth, so that their state does not change and we can examine them with the effects of microgravity still in their system. Clamp B would be released halfway through the 4-week period. It would then allow the planarian to have access to their food. Clamp A, which holds the chamber containing neutral buffered formalin, would be released 2 days before the planarian are sent back to Earth.
We performed several experiments on Earth to gather control information and ground truth to compare to the data that we would collect on the ISS. Firstly, we tested to see if our planaria would regenerate if cut in half, not through asexual reproduction. We used a scalpel and cut several worms in half, observing them over the course of a week. The first few worms died due to inadequate habitats, but one of the worms successfully regenerated into 2 whole separate worms. Secondly, we ran a test that would show us if planaria are capable of surviving for extended periods of time in a confined space. We placed 3 worms in a test tube with about as much water as they would be in on the ISS and observed them over a week. One planarian, though, due to lack of proper food, resorted to cannibalism and ate the other 2 worms. Even though this did show a potential setback if we were to send the worms to space, the remaining planaria did not die for another week or so, proving adequate enough for an experiment in such an environment. As our final hypothesis, we hypothesized that due to our understanding of cellular atrophy in humans in a microgravity environment, the planarian will not be able to regenerate in microgravity as opposed to its proven ability to do so in gravity. We decided this because human muscles have been proven to atrophy in microgravity due to the slowed activity of the tissue-specific stem cells. Since planaria depend on their stem cells for their regeneration process, we think that their ability to regenerate will be slowed or possibly stopped by this change.
We would like to thank the NCE SSE and our school, Kent Place School, for giving us the opportunity to speak to you all today. Thank you.